Hello and welcome. Let's start today by talking about Chapter 8, Photosynthesis. This is a very important chapter, just like cellular respiration was. Now here when we go through and we talk photosynthesis, here photosynthesis you'll see is going to be responsible for converting solar energy into chemical energy of a carbohydrate. Photosynthetic organisms such as plants, and we'll see in this chapter our focus is going to be on flowering plants is what we're going to be looking at. So here we could see photosynthetic organisms such as plants, algae, and cyanobacteria are called autotrophs. They're called autotrophs because they produce their own food. Now here in figure 8.1, we could see number one, we've got a bacterium. This bacterium is Oscillatoria. It's a type of cyanobacteria. And then second, we can see in here a type of algae. This is kelp. Kelp can range from microscopic to macroscopic. And then third, here we can see plants such as sequoia trees. So again here, our focus is going to be on flowering plants. These are all various types of photosynthetic organisms. So here, when we go through and we talk about photosynthetic organisms, I'd like you to know each year photosynthesizing organisms are going to produce between 100 and 200 billion metric tons of carbohydrates. Let me repeat that. Each year, photosynthesizing organisms produce between 100 and 200 billion metric tons of carbohydrates. This demonstrates how they're able to sustain not only themselves, but then all other organisms found on Earth also. Now, most food chains trace back to plants and algae. These producers have the ability to synthesize carbohydrates, which feeds them, we said, and consumers too. When we talk consumers, consumers are also known as heterotrophs. They're also known as heterotrophs, and they must take in preformed organic molecules. Both use organic molecules produced by photosynthesis as a source of building blocks for growth, repair, and as a source of chemical energy for cellular work. Pigments are going to allow photosynthetic organisms to capture solar energy. Pigments will allow photosynthetic organisms to capture solar energy, which fuels photosynthesis. Most photosynthetic organisms contain chlorophyll, the pigment that gives them a green color. Some contain carotenoids, which gives photosynthesizing cells a yellow to red color. And then last, we have phycobilins. Phycobilins are going to give red algae their red color and cyanobacteria a bluish color. Portions of plants, particularly the leaves, contain chlorophyll and other pigments. The leaf of a flowering plant contains mesophyll tissue, which contains cells that are specialized for photosynthesis. So here we could see that. We'll jump a little forward right to here. Here we've got a leaf. When we look at that leaf in greater detail then, we can move to the next picture. Here we're able to appreciate the mesophyll of the leaf. And then here we've got the upper epidermis. Here we have the lower epidermis. And then here you can see we've got the cuticle. When we focus on these mesophyll cells in greater detail, here we're going to be able to see then, let me go back in here. Here inside these mesophyll cells, we've got these chloroplasts. So here when we zoom in on these mesophyll cells, we're going to be able to appreciate chloroplasts. So here you could see we've got chloroplasts. And then inside the chloroplast, we're able to appreciate other structures that we're going to go through and we're going to talk about as well. So again, the leaf of a flowering plant contains mesophyll tissue, which contains cells that are specialized for photosynthesis. So these are those mesophyll cells. The raw material for photosynthesis we'll see is going to be water and carbon dioxide. The roots of a plant absorb water, which then moves through vascular tissue up a stem to a leaf by way of leaf veins. Carbon dioxide from the air enters a leaf through small openings we saw here that are going to be called stomata. Here's those tiny openings that are going to allow carbon dioxide in, and eventually we'll see oxygen out. So carbon dioxide, we can see, enters through stomata. The air and water diffuse into the cells and enter chloroplasts, the organelles that are going to be responsible for carrying out photosynthesis. Again, here are the beautiful chloroplasts. A double membrane surrounds a chloroplast and its fluid-filled interior. So here you can see the double membrane, inner and outer membrane, that are going to surround the chloroplast and its fluid-filled interior, which is called the stroma. The stroma. A different membrane system within the stroma forms flattened sacs, we can see there, that are called thylakoids. Thylakoids. In some places, you can see these thylakoids are stacked to form then what we call grana. Singular, you can see granum. With all these grana right inside of here. The spaces of each thylakoid is connected to the space of every other thylakoid. So here you can see that in this picture. The space of each thylakoid is going to be connected to the space of every other thylakoid within a chloroplast, forming then a continuous inner compartment within the chloroplast called a thylakoid space. So these channels between the thylakoids are going to allow each of the thylakoids to then communicate with each other, forming what we call thylakoid space. Chlorophyll and other pigments that are part of the thylakoid membrane are capable of absorbing solar energy. They are capable of absorbing solar energy. This is the energy that drives photosynthesis. This is the energy that drives photosynthesis. The stroma is filled with enzymes, and it is the site where carbon dioxide is first attached to an organic compound, and it is then reduced to a carbohydrate. Humans, and indeed all organisms, they release carbon dioxide into air as a waste product from metabolic respiration, such as cellular respiration, as we saw in the previous chapter. This is some of the carbon dioxide that enters a leaf through the stomata and gets converted to a carbohydrate. Carbohydrate in the form of glucose. Okay, carbohydrate in the form of glucose. We'll see it's the chief energy source for most organisms. Carbohydrate in the form of glucose, because we'll see this is the chief energy source for most organisms. And that's why we saw carbohydrate metabolism, basically cellular respiration, right, in relation to glucose. We started with glucose, and we wound up with, in the first part, pyruvate, and we saw that pyruvate eventually keep going further into oxaloacetate. And then all of this NADH plus hydrogen ions being released, helping to then create ATP. So here we can see the photosynthetic reaction right inside of here. Here in this photosynthetic reaction, so here we can appreciate how 6 carbon dioxide and 6 water will react along with solar energy and with the various pigments we saw to help form C6H12O6, which we said is glucose, plus 6 oxygen molecules. So let's jump back here and let's watch this short video.
Within eukaryotic photosynthetic organisms, also known as photoautotrophs, the chemical reactions of photosynthesis occur within plant cells in specialized structures known as chloroplasts. Photosynthesis consists of two sets of reactions, the light-dependent reactions and the Calvin cycle. Within the chloroplast are small disc-like structures called thylakoids, which are surrounded by a fluid-filled space called the stroma. The reactions that synthesize glucose, the Calvin cycle, occur in the stroma. The light-dependent reactions occur in the thylakoid. It is here that conversion of light energy to chemical energy is initiated.
All right. Now, this oxygen, the oxygen given off by photosynthesis comes from water. It comes from water, and this was proven experimentally. Photosynthesis tells us why plants are so important to our lives. So I feel this is a very important chapter. Plants are going to provide us with food, as we can see now, in the form of carbohydrates. Plants provide us with food in the form of carbohydrates and much of the oxygen that we breathe and use for cellular respiration. And we saw how important oxygen was for cellular respiration, especially when we talked about Krebs cycle and the electron transport chain. Let's talk about then the two sets of reactions. Let's look at photosynthesis in greater detail now. When we talk photosynthesis, it suggests that the process requires two sets of reactions. Photo first, which means light, and it refers to the reactions that capture solar energy. And second, then we have the term synthesis. Synthesis, it refers to the reactions that are going to produce carbohydrates. The two sets of reactions are called light reactions, or we can call them light-dependent reactions. The reason why we call them light-dependent reactions because they cannot occur unless light is present. And then second, we have our Calvin cycle reactions, which we can also refer to as our light-independent reactions, because they will occur whether or not light is present. So here we can see that inside the chloroplast then, we're able to appreciate our light reactions and then our light-independent reactions, Calvin cycle reactions. And we'll see then how they're all coming together to form oxygen and our carbohydrate. We will see that the light reactions release oxygen and provide the molecules that allow the Calvin cycle reactions to reduce carbon dioxide to a carbohydrate. The coenzyme NAD+ we see here now, nicotinamide adenine dinucleotide phosphate, carries hydrogen atoms from the light reactions to, you can see, the Calvin cycle reactions. When NAD+ accepts hydrogens, it becomes NADPH. Also, we can see here ATP. ATP is going to carry energy. It carries energy from the light reactions to our Calvin cycle reactions. Next, then, let's look at plants as solar energy converters. So this will take us to then 8.2. Now, when we talk about plants as solar energy converters, I want you to know during the light reactions, various pigments within the thylakoid membranes absorb solar energy. Solar energy or radiant energy from the sun can be described in the terms of its wavelength and its energy content. Figure 8.4 we can see here. Figure 8.4, it lists the different types of radiant energy. It lists the different types of radiant energy from, we can see, the shortest wavelengths, which are gamma rays, to our longest wavelengths, which are radio waves. So in between the two, you can see we have X-rays, UV, visible light, infrared, and microwaves. X-rays obviously help us to detect fractures and various other problems. UV we need for vitamin D conversion. Too much, bad for us. Too much X-rays, bad for us. Visible light is one part of the spectrum that our eyes respond to. Infrared waves we have, microwaves, and obviously we know there as well, and then radio waves. Now, when we talk about visible light, white or visible light is only a small portion of the spectrum. So we can see white light or visible light is only a small portion of the spectrum. Visible light itself contains various wavelengths of light. When visible light is passed through a prism, we'll see it's going to be dispersed. We're going to see all the different colors that make up visible light. The sensory areas of our brains then identify these wavelengths as distinct colors. Now, the colors in visible light range from violet, which is going to be the shortest wavelength, to indigo, blue, green, yellow, orange, and red, which is going to be the longest wavelength. Right? In kindergarten now, these kids, when I was in kindergarten, we were barely able to learn our few colors. Kids nowadays have a mnemonic for that even, and they refer to that mnemonic as Roy G. Biv. Red, orange, yellow, and they go through each letter. And my daughter was playing these videos from YouTube, and they were singing Roy G. Biv, Roy G. Biv. I'm like, wait, why is she learning about mnemonics? She's barely in kindergarten. But again, it's an easy way to remember information. The pigments found within most types of photosynthesizing cells, the chlorophyll A and B, and then also the carotenoids, they're capable of absorbing various portions of visible light. You can see here the absorption spectrum, and here we can see both chlorophyll A and B, they absorb violet, indigo, blue, and red light better than light of other colors. Better than the light of other colors. Green light is reflected and only minimally absorbed, making leaves appear green to us. So you can see yellow and orange carotenoids are able to absorb light in the violet, blue, green range. In the violet, blue, green range. These pigments become noticeable in the fall when chlorophyll breaks down and other pigments are uncovered. Photosynthesis begins when the pigments within the thylakoid membrane absorb solar energy. Let's move to our then light reactions. Let's talk about our light reactions. So before we jump forward, again, I want you to remember green light is reflected and only minimally absorbed. That's why leaves appear green. Same thing with grass. Okay, if you're wearing a red shirt, same thing would be with red. Red light is going to be reflected and only minimally absorbed, making the red shirt appear red. Next, then, when we talk light reactions, the light reactions that occur in the thylakoid membrane consist of two electron pathways that are going to be called the non-cyclic electron pathway and the cyclic electron pathway. Both electron pathways produce ATP, but only the non-cyclic pathway also produces NADPH. Let's look at then first the non-cyclic electron pathway, and then we'll jump over to the cyclic electron pathway. But before we do that, we'll check out this quick video about photosynthesis.
In order for plants to grow, they need inputs of carbon dioxide, water, and energy. The chemical process by which plants use these resources to manufacture glucose, the building blocks of plants, is called photosynthesis. In the process, oxygen gas is produced as a byproduct. The energy for photosynthesis originates in the sun and arrives at the earth as sunlight. This light has both a wave and a particle nature. The particles, or photons, are the smallest units of light. Photons oscillate along a path, which is measured as wavelengths. The light emitted from the sun contains photons in a wide spectrum of wavelengths, called the electromagnetic spectrum. Photosynthetic organisms use only a small portion of the electromagnetic spectrum, called visible light. Photosynthetic organisms contain pigments that facilitate the capture of wavelengths of light in the visible light range. The color of the pigment comes from the wavelengths of light reflected. Plants appear green because they reflect yellow and green wavelengths of light. Red and blue wavelengths of light are absorbed by these pigments and provide the energy that is used for photosynthesis.
So moving forward, then let's talk about the non-cyclic electron pathways. Now, the non-cyclic electron pathway is so named because the electron flow can be traced from water to a molecule of NAD+. So here in this figure 8.6, we can see this pathway uses two photosystems. Those photosystems are called photosystem 1. You can see right inside of here, PS1, photosystem 1, and photosystem 2, PS2, photosystem 2, we could see here. They are named for the order in which they were discovered, not in the order in which they participate in the photosynthetic process. A photosystem consists of protein complex, chlorophyll A, chlorophyll B, carotenoids, and an electron acceptor. So here you've got in photosystem 1, pigment complex, and then we've got our electron acceptor. The non-cyclic pathways begin with photosystem 2. The pigment complex absorbs solar energy, which is then passed from one pigment molecule to another until it is concentrated in a particular pair of chlorophyll A molecules that are called the reaction center. Electrons in the reaction center chlorophyll become so energized that they escape from the reaction center and move to a nearby electron acceptor. Photosystem 2 would disintegrate without replacement electrons. These replacement electrons are supplied by water. They're provided by water, which splits, releasing oxygen to the atmosphere. Many organisms, including plants and even ourselves, we use this oxygen. The hydrogen ions are going to stay in the thylakoid space, and they are going to contribute to the formation of a hydrogen ion gradient. And they're going to contribute to the formation of a hydrogen ion gradient. As shown in the overall chemical reaction for photosynthesis, water is going to be used up and oxygen is going to be produced. The electron acceptor that received the energized electrons from the reaction center will send those electrons now down a series of carriers that are going to pass the electrons from one to the other, all the way through, and they're going to be known as the electron transport chain, the E ETC, the electron transport chain. So as the electrons pass from one carrier to the next, the energy that is released is going to be used to move hydrogen ions from the stroma into the thylakoid space, helping to form a hydrogen ion gradient like we saw with cellular respiration. And then again, when these hydrogen ions flow down their electrochemical gradient through ATP synthase complexes, ATP production is going to occur. This ATP will be used in the Calvin cycle reactions in the stroma to reduce carbon dioxide to a carbohydrate. Similarly, when the photosystem 1 pigment complexes absorb solar energy, energized electrons leave its reaction center and are captured by a different electron acceptor. So now, looking back, after going through the electron transport chain, the electrons from photosystem 2 are now, then you can see, low energy electrons because they have given up their energy to, we said, the carriers. These electrons are used to replace those lost by photosystem 1. The electron acceptor here in photosystem 1 then is going to pass its electrons off onto the NADP plus molecules. Each NADP plus molecule we can see here then accepts 2 electrons and a hydrogen ion to become the reduced form of the molecule called NADPH. This NADPH will also be used by the Calvin cycle reactions in the stroma to reduce carbon dioxide to a carbohydrate. So let's look at this organization of the thylakoid membrane. Now, the following molecular complexes are going to be present in the thylakoid membrane and are involved in the non-cyclic electron pathway. So here we could see, we could move over to figure 8.7. So here we have photosystem 2, our electron transport chain, and then photosystem 1. So photosystem 2, which consists of a pigment complex and an electron acceptor molecule, receives electrons from water, which splits, then releasing oxygen, we said. The electron transport chain, it's responsible now for carrying electrons from photosystem 2 to photosystem 1, and it pumps hydrogen ions, from the stroma to the thylakoid space, as we mentioned earlier as well. Photosystem 1, which consists of a pigment complex and electron acceptor molecules, is adjacent to, you can see, the NAD plus reductase, which reduces NAD plus to NADPH, we said. The ATP synthase complex, you can see here now, the ATP synthase complex, it crosses the thylakoid membrane. We can see that here very nicely. It crosses the thylakoid membrane and it contains an interior channel and a protruding ATP synthase. This is an enzyme that helps to join ADP to a phosphate, creating ATP, which we refer to as chemiosmosis. Let's look at then the cyclic electron pathway. So when we talk about the cyclic electron pathway, now here I'd like you to know, under some environmental conditions, such as high oxygen levels, NADPH levels may accumulate in the cell, and therefore photosynthetic cells may enter the cyclic electron pathway. Here we can appreciate that here in figure 8.8. This pathway is also found in many prokaryotic cells. It differs from the non-cyclic reactions in that the electrons are recycled back to photosystem 1. The electrons you see here are recycled back to photosystem 1. The pathway begins when photosystem 1 pigment complex absorbs solar energy that's passed from one pigment to the next until it becomes concentrated in a reaction center. As with photosystem 2, the electrons become so energized that they escape the reaction center and they move up to a nearby electron acceptor molecule. This time, instead of the electrons moving on to NADP+, energized electrons are taken up by an electron acceptor and are sent down an electron transport chain. As the electrons pass from one carrier to the next, their energy becomes stored as hydrogen ion gradient. Again, the flow of hydrogen ions down their electrical chemical gradient through ATP synthase complexes produces ATP. Again, the flow of hydrogen ions down their electrochemical gradient through ATP synthase complexes produces ATP. We've seen that. The spent electrons, they return to photosystem 1 after the electron transport chain. This is how photosystem 1 receives replacement electrons and why this electron pathway is called cyclic. It is also why the cyclic pathways produce ATP but do not produce NADPH. It is also why the cyclic pathways produce ATP but does not produce NADPH. Let's talk ATP production. Now, when we talk ATP production, the thylakoid space acts as a reservoir for hydrogen ions. Each time oxygen is removed from the water, 2 hydrogen ions remain in the thylakoid space. As the electrons move from carrier to carrier along the electron transport chain, the electrons give up energy, which is used to pump hydrogen ions from the stroma into the thylakoid space. This is like pumping water into a reservoir. The end result is that there are more hydrogen ions then in the thylakoid space than in the stroma. Because hydrogen ions have a charge to them, this is an electrochemical gradient. Not only are there more hydrogen ions, but there are more positive charges within the thylakoid space than the stroma. When a channel is opened in the ATP synthase complex, hydrogen ions, these protons, are going to flow from the thylakoid space into the stroma. Just like water flowing out of a reservoir. The flow of hydrogen ions from high concentration to low concentration across the thylakoid membrane provides the energy that allows the ATP synthase enzyme to enzymatically produce ATP from ADP and a phosphate. This method of producing ATP, I told you, is going to be called chemiosmosis. Because ATP production is tied to the establishment of a proton gradient. Let's move over to 8.3 then. Now when we move over to 8.3, before we do that, let's watch this video here. We have a couple of videos. Let's watch both of these videos and then we'll move forward.
When glucose is oxidized during glycolysis and the citric acid cycle, NAD+ and FAD are reduced to NADH and FADH2, and hydrogen.
This was electron transport synthesis
ions,
we saw.
H+, are released. In the mitochondria, the electrons from NADH are transferred to the electron transport proteins, and the hydrogen ions are transferred across the membrane. As the electrons move from carrier to carrier down the electron transport chain, more hydrogen ions are carried across the membrane. The terminal electron acceptor for this process is oxygen. When oxygen is reduced, water is produced. The transfer of hydrogen ions generates a potential energy gradient across the membrane of the mitochondrion. Because membranes are impermeable to ions, the hydrogen ions that re-enter the matrix must pass through special proton channel proteins called ATP synthase. The energy derived from the movement of these protons is used to synthesize ATP from ADP and phosphate by the ATP synthase complex.
Again, so that was just a review of the electron transport chain we saw in cellular respiration.
Proton pumps are protein complexes that move hydrogen ions across cell membranes. As protons move through the proton pump, they begin to build up on one side of the membrane. As a result, a concentration gradient of hydrogen ions that contains potential energy is formed across the membrane. The membrane is not permeable to the charged hydrogen ions, and they cannot diffuse back across the membrane. Instead, they must pass through a special channel. The protons move through this channel, which is the enzyme ATP synthase. Energy is released as the hydrogen ions move down their concentration gradient. The movement of hydrogen ions down a concentration gradient provides the energy for ATP synthase to form ATP from ADP and phosphate.
So we've seen this proton pump now twice. We saw it with the electron transport chain, with the video we just saw, and then we've seen it here as well in relation to photosynthesis. Here we saw it here in our thylakoid membrane. So right here we could see our hydrogen pumps are bringing hydrogens, we said, from the stroma into the thylakoid space where we can see this concentration gradient being built. And then here we've got our ATP synthase that's going to allow these protons to make their way out. So let's move over to 8.3. Now 8.3 deals with plants as carbon dioxide fixers. So now when we look at 8.3 and we talk about plants as carbon dioxide fixers, the Calvin cycle reactions take place after the light reactions. This is a series of reactions that produces a carbohydrate before returning to the starting point. So here we can move to figure 8.9. Here we can see the cycle is named for Melvin Calvin, who, with others, discovered the reactions that make up the cycle. This series of reactions uses atmospheric carbon dioxide to produce a carbohydrate. Humans and most other organisms take in oxygen from the atmosphere and release carbon dioxide. The Calvin cycle, it includes, number 1, we'll see carbon dioxide fixation. Number 2, carbon dioxide reduction. And number 3, the regeneration of RUBP, ribulose 1,5-bisphosphate, which is the starting material of the cycle. Carbon dioxide fixation is the first step of the Calvin cycle. During this reaction, 3 molecules we can see right inside of here of carbon dioxide, 3 molecules of carbon dioxide from the atmosphere are going to be attached to 3 molecules of RUBP, which is a 5-carbon molecule. The result is 3 6-carbon molecules. Each 6-carbon molecule then splits in half, forming a total of 6 3-carbon molecules. The first 3-carbon molecule is called 3-phosphoglycerate, or 3PG. The enzyme that speeds this reaction is going to be called RUBP carboxylase. It is a protein that makes up about 20 to 50% of protein content in chloroplasts. Next, let's look at the reduction of carbon dioxide. Each of the two 3PG molecules undergoes reduction to G3P, and that's going to happen in 2 steps. The first step, we'll see, the first step is going to convert our 3PG molecule into 1,3-bisphosphoglycerate, or into BPG, we can abbreviate it as. And this is going to happen by the use of ATP molecules. Look at here, 6 ATP molecules are going to be used. Then in the second reaction is going to convert BPG, our bisphosphoglycerate, into glyceraldehyde 3-phosphate, into G3P. And that will happen by the use of NADPH, 6-NADPH to be exact. This sequence of reactions uses ATP and NADPH from the light reactions that we formed. These reactions also result in the reduction of carbon dioxide to a carbohydrate molecule. These reactions also result in the reduction of carbon dioxide to a carbohydrate because an R group attached to CO2 has now become an R group attached to a CH2O. Energy and electrons are needed for this reduction reaction, and they are supplied by ATP and NADPH. Let's look at then the regeneration of RUBP. But before we do that, we'll check this video out real quick.
In most photosynthetic organisms, thylakoids contain pairs of photosystems, called photosystem 1 and photosystem 2, that work in tandem to produce the energy that will later be used in the stroma to manufacture sugars. The photosystems of the thylakoid consist of a network of accessory pigment molecules and chlorophyll, the molecules that absorb the photons of light. Within the pigment molecules, the absorbed light energy excites electrons to a higher state. Photosystems will channel the excitation energy gathered by the pigment molecules to a reaction center chlorophyll molecule, which will then pass the electrons to a series of proteins located on the thylakoid membrane. Photons of light strike photosystems 1 and 2 simultaneously. We will examine what happens with the photons striking photosystem 2 first. The energized electrons are passed from the reaction center of photosystem 2 to an electron transport chain. The electrons lost by photosystem 2 are replaced by a process called photolysis, which involves the oxidation of a water molecule producing free electrons and oxygen gas. While this oxygen gas is a byproduct of photosynthesis, it is an important input to the cellular respiration pathways. As electrons pass through the electron transport chain, the energy from the electron is used to pump hydrogen ions from the stroma to the thylakoid, creating a concentration gradient. This gradient powers a protein called ATP synthase, which phosphorylates ADP to form ATP. The low-energy electrons leaving photosystem 2 are shuttled to photosystem 1. Within photosystem 1, low-energy electrons are re-energized and are passed through an electron transport chain where they are used to reduce the electron carrier NADP+ to NADPH. When the chloroplast is receiving a steady supply of photons, NADPH and ATP molecules are rapidly being provided to the metabolic pathways in the stroma. Therefore, the ATP and NADPH formed during the light-dependent reactions are used in the stroma to fuel the Calvin cycle reactions. The Calvin cycle consists of a series of reactions that reduce carbon dioxide to produce the carbohydrate glyceraldehyde 3-phosphate. The cycle consists of 3 steps, the first of which is carbon fixation. In this step, carbon dioxide is attached to ribulose 1,5-bisphosphate, resulting in a 6-carbon molecule that splits into 2 3-carbon molecules. The second step is a sequence of reactions using electrons from NADPH and some of the ATP to reduce carbon dioxide. In the final step, ribulose 1,5-bisphosphate is regenerated. For every 3 turns of the cycle, 5 molecules of glyceraldehyde 3-phosphate are used to reform 3 molecules of ribulose 1,5-bisphosphate. The remaining glyceraldehyde 3-phosphate is then used to make glucose, fatty acids, or glycerol. It takes 2 molecules of glyceraldehyde 3-phosphate to make 1 molecule of glucose phosphate. Thus, the Calvin cycle has to run 6 times to produce 1 molecule of glucose. These molecules can remove their phosphate and add fructose to form sucrose, the molecule plants use to transport carbohydrates throughout their system. Glucose phosphate is also the starting molecule for the synthesis of starch and cellulose.
I'd like you to notice that the Calvin cycle reactions we see here in figure 8.9 are multiplied by 3 because it takes 3 turns of the Calvin cycle to allow 1 G3P to exit. So for every 3 turns of the Calvin cycle, 5 molecules of G3P are used to reform 3 molecules of RUBP, and the cycle continues. Notice that 5 times 3, which is the carbons in G3P, is going to be equal to 3 times 5, which is the carbons in RUBP. So here we could see that. So we saw that reaction in greater detail, 2-step process. And then here you could see 5 times 3 carbons in our G3P is going to be equal to 3 times 5, which is carbons in our RUBP. This reaction also uses some of the ATP produced by the light reactions. It also uses some of the ATP that's produced by the light reactions. Let's talk about the importance of the Calvin cycle. When we talk about the importance of the Calvin cycle, we'll see G3P or glyceraldehyde 3-phosphate is the product of the Calvin cycle that can be converted to other molecules a plant needs. Compared to animal cells, algae and plants have enormous biochemical capabilities. They use G3P for glucose, sucrose, starch, cellulose, fatty acids, and amino acid synthesis. Glucose phosphate is one of the organic molecules that results from G3P metabolism. Glucose is the molecule that plants and animals most often metabolize to produce the ATP molecules that they require for their energy needs. And again, we saw that in the previous chapter. Glucose phosphate can be combined with fructose and the phosphate removed to form sucrose, the molecule plants use to transport carbohydrates from one part of the plant to another. Glucose phosphate. As we saw, glucose phosphate is also the starting point for the synthesis of starch and cellulose. Starch is a storage form of glucose. Some starch is stored in chloroplasts, but most starch is stored in the roots. Cellulose is a structural component of plant cell walls and becomes fiber in our diet because we're unable to digest it. A plant can use the hydrocarbon skeleton of G3P to form fatty acids and glycerol, which are combined in plant oils such as corn oil, sunflower oil, or olive oil, commonly used in cooking. So here we've got our non-cyclic pathways we can check out, and then we'll check out our Calvin cycle reactions. And then here you can see we also have basically alternate pathways for photosynthesis. We won't get into that, but if you'd like, you're welcome to check them out. I included them in here. C3 photosynthesis, which is going to be basically occurring in wheat, rice, and oats. And then also C4 photosynthesis, which will occur in sugarcane and corn. And then here we can see chem photosynthesis, which is going to be the most prevalent pathway amongst succulent plants that grow in the desert, including cacti. So here you've got chem photosynthesis. Let's check out our last few videos and then we'll...
In this diagram, we are looking at a simplified schematic of the types of reactions that occur within the chloroplast of a photosynthetic cell. The inputs shown here, water, carbon dioxide, and solar energy, or sunlight, are all coming from outside of the cell. We are going to focus on the light reactions, which are the first step of the photosynthetic process in most plants. As we proceed through these diagrams, notice how these reactions have inputs of solar energy and water. In order to understand the light reactions, it is often easiest to focus on the flow of electrons, shown here as blue circles. Let's start by exploring what is happening in photosystem 2. Photosystems are collections of pigments that concentrate the incoming solar energy on a reaction center. This energy causes the release of high-energy electrons, which in turn are collected by an electron acceptor. The high-energy electrons, which contain the energy originally found in the incoming photons, are then passed to the electron transport chain, or ETC. The ETC harvests the energy from these electrons and use it to charge adenosine diphosphate, or ADP molecules, forming adenosine triphosphate, or ATP. Remember that this is a non-cyclic reaction, so these electrons are not returned to photosystem 2. Instead, they are handed off to photosystem 1. At the same time as these reactions are occurring in photosystem 2, sunlight is also striking photosystem 1, causing a similar release of high-energy electrons. However, unlike the electrons from photosystem 2, which were used to generate ATP, these electrons are captured to reduce NADP+, forming NADPH. So, as you can see, this set of reactions is not cyclic, meaning that the electrons are not returned to where they started from. The electrons leaving photosystem 2 are used as replacement electrons for photosystem 1, while the electrons from photosystem 1 are used to form NADPH. However, there is one small problem. Over time, photosystem 2 would lose all of its electrons and disintegrate. But this does not happen due to the fact that some of the solar energy in photosystem 2 is used to split molecules of water, releasing electrons. This is called photolysis. The electrons being generated during photolysis replenish the electrons being lost in photosystem 2. Notice how some oxygen is also produced. This oxygen exits the cell as oxygen gas. Finally, we can take a look at what happens to the ATP and NADPH produced during the light reactions. Remember that this is only the first stage of photosynthesis, and that the overall goal was to capture the energy of the sun and produce carbohydrates. In this first set of reactions, we have captured solar energy and stored it temporarily as ATP and NADPH. Both of these molecules will now be used in the Calvin reactions in the formation of carbohydrates, or CH2O. Once used, they are recycled back to the light-dependent reactions as ADP and NADP+, respectively. Photosynthesis is a two-step process. The first set of reactions, called the light reactions, transfer the kinetic energy of sunlight into the molecules ATP and NADPH. In the second set of reactions, we are going to use this energy in carbon dioxide gas from the atmosphere to produce carbohydrates. As you proceed through the stages of the Calvin cycle, do not lose sight of the fact that the overall goal is to produce carbohydrates. The Calvin cycle begins with an input of 3 molecules of carbon dioxide, or CO2. Carbon dioxide is a gas, so to facilitate the following chemical reactions, it is combined with an organic molecule. That molecule is called RUBP, and the process is called carbon fixation. Carbon fixation produces a 6-carbon molecule, which shortly thereafter splits to form a pair of 3 carbon molecules called 3PG. The next stage of the Calvin cycle uses the energy products made during the light reactions to reduce the 3PG molecules. This stage requires an input of energy, which is supplied by ATP. Remember that reduction involves an addition of electrons, which in the Calvin cycle is supplied by NADPH. The addition of the electrons from NADPH reduces 3PG to form G3P. The ADP and NADP+ from this stage return to the light reactions to be recharged. G3P is an important intermediate of the Calvin cycle, since it is the precursor for the formation of many organic molecules, including carbohydrates such as glucose. At this stage, the plant has successfully harvested the energy from the sun and used it to reduce carbon dioxide gas to form carbohydrates. However, this process would be relatively inefficient if it were not possible to recycle some of the intermediate molecules, specifically RUBP. The last step of the Calvin cycle acts as its recycling phase. Some of the G3P produced earlier is modified using ATP, which originated in the light reactions. The RUBP produced by this stage may be used to fix more CO2, thus continuing the cycle.